In this video lecture, we will discuss about the phosphatase test. Why this test it is been used here. This test it is been used to check the quality of an pasteurized milk. Pasteurized milk which is safe for the consumption of humans too. We can take it or we can consume it easily in the safe way where it can kill the bacteria or harmful bacteria. What is phosphatase? Phosphatase is an enzyme in which the phosphate group is been removed. This enzyme removes the phosphate group. That's why it is known as an phosphatase test. Here the alkaline phosphatase is been used to check the quality of the milk. Now, what is introduction? Alkaline phosphatase. Alkaline phosphatase is an enzyme which is naturally present in the milk. Here the alkaline phosphatase, it is an enzyme which is naturally present in the unpasteurized milk or milk which is not been pasteurized. Here this enzyme denatures at the pasteurization. Here this enzyme is been denatured at the pasteurization temperature. If milk is properly pasteurized, then alkaline phosphatase is absent. If our milk sample or milk is been properly pasteurized, that means here there will be the absence of the alkaline phosphatase enzyme, because the alkaline phosphatase enzymes will denature or destroy at the pasteurization temperature. If milk is not properly pasteurized, then the alkaline phosphatase is present in its active form. Here the milk, if the milk is not pasteurized properly, the alkaline phosphatase will be present in its active form. What is alkaline phosphatase enzyme? How does it works or it functions? The phosphatase means the removal of phosphate group from the product, and alkaline means which will be active in the alkaline condition. So it is named as an alkaline phosphatase. Here further, there are many enzymes which are present in the milk which is used to check the quality of milk, such as calamase, which is protease, lactase. All these are the enzymes which are also present in the milk sample. But here the alkaline phosphatase has been used because to determine or to check the quality of milk, because the alkaline phosphatase it is destroyed or denatured at the pasteurization temperature. But all these enzyme mentioned here, they get denatured at the temperature below the pasteurization. The pasteurization temperature is 71 degrees Celsius or above. Here these all enzymes such as like calamase, lactase and protease enzymes which can be destroyed or denatured below or lower than the pasteurization temperature. So these all enzymes are not considered to check the quality of the milk. So there is no need to check the presence or the absence of the enzymes. Here these enzymes are not been considered to check the quality of milk. Here only alkaline phosphatase which can be destroyed or denatured at the pasteurization temperature, while other enzymes are denatured or destroyed below the pasteurization temperature. Now we will see about the principle. On what principle does this enzymes or this test works? Here, first of all, the presence of an enzyme can be determined by the production of the product from the substrate. Here this enzymes is been produced or determined by the production of the product. Here the substrate from which the product is been formed. Here as a substrate we have taken the para nitrophenyl phosphate. Here as a substrate we can take as an para nitrophenyl phosphate in the for the enzyme alkaline phosphatase. Alkaline phosphatase which can, here we can use as a substrate as a para nitrophenyl phosphate. Now this substrate will convert into the product. Now para-nitrophenyl phosphate is converted into para-nitrophenol and phosphate is converted into two products. First one is an para-nitrophenol and another one is an phosphate. Here the para-nitrophenol is an yellow colored product which can be determined through this color intensity. We can determine the concentration of alkaline phosphatase present in our milk sample. Here as an substrate we have taken para-nitrophenyl phosphate, which is converted into two main products. First one is para-nitrophenol, which is yellow color product, and another one is an phosphate which is been separated here because the enzyme name is an phosphatase, which can separate the phosphate group or remove the phosphate group from the product. Here the para-nitrophenol is yellow color product. Here para-nitrophenyl is an yellow color product whose intensity is measured at 405 nanometer. Here the intensity of color or color intensity which is obtained from the product, which is an para-nitrophenol, which is an product which gives an yellow color formation or yellow color product, which can be those concentration or intensity which it is been measured at the 405 nanometer in the colorimeter. Here we can see the increase in the intensity of yellow color is directly proportional to the amount or concentration of alkaline phosphatase present in the milk. Here what we will indicate if the intensity or the concentration of yellow color will be in the high amount. If the concentration or amount of yellow color will be high in the higher amount, that will indicate that the milk is not pasteurized well and the presence of an alkaline phosphatase enzyme in the milk sample. What are the requirements? Now we will see how this test is been performed. For that we will need requirement. What type of requirements we will need? First one is an carbonate buffer. First one is an carbonate buffer which maintains the alkaline condition in the test, because the enzyme itself indicates the alkaline phosphatase. This enzyme will only be activate in the alkaline condition. The second one is an para-nitrophenyl phosphate, in which concentration 0.01 molar concentration, which will act as an substrate here. And another one is an milk sample, glass vase and colorimeter. This all are the requirements needed in our to perform the phosphatase test. Now we will see how this test is been performed. First of all, take 2 ml of the milk sample. In the tube 0.05 ml of substrate, which is an para-nitrophenyl phosphate, plus 0.5 ml of the buffer solution. Here we will take the 3 tubes. First one is standard, another one is an test, and third one is an blank. Here we will, in the standard tube we will not take a milk sample, while in the blank we will only add the distilled water. In the standard we will only add the substrate and the buffer solution, while in the blank we will only add the milk sample and the buffer solution. Here in the blank tube we will only indicate whether it is act as an control. Blank will act as a negative control, while standard will act as an positive control. In the test tube we will add 2 ml of milk sample, 0.05 ml of para-nitrophenyl phosphate and 0.5 ml of the buffer solution, while in the standard we will only add the substrate plus buffer, while in the blank we will only add milk sample or distilled water plus buffer. In blank we will only add distilled water and buffer solution. In blank we will not add an substrate which is an para-nitrophenyl phosphate, but in the test labeled tube we will add milk sample, substrate as well as an buffer solution. Next, in all these three tubes will be incubated at the room temperature for the 10 minutes. Here all the three tubes is been incubated at room temperature for the 10 minutes. Here after the incubation the intensity of the yellow color is been measured at 405 nanometer in the colorimeter. Here after the incubation of 10 minutes at room temperature we will measure the intensity of yellow color at 405 nanometer in the colorimeter, and at the end note down the results. Now we will see on what basis you will interpret or you will conclude the results. Here the after the 10 minutes if yellow color is developed, it is clearly indicates the presence of an alkaline phosphatase enzyme present in the milk. After the 10 minutes if your tubes contain yellow color, that will be clearly indicate there will be the presence of an alkaline phosphatase enzyme which is present, which will indicate that your milk sample is not properly pasteurized or it is not safe for the consumption of the humans. Here in the place of substrate, here we can take another substrate. Here the substrate which is an para-nitrophenol phenyl phosphate which gives an yellow color. If the intensity of that yellow color is in higher intensity or in the higher amount, it will indicate that the presence of alkaline phosphatase in that milk sample. Now what are the disadvantages, or what if the alkaline phosphatase can be consumed or in way consumed by the humans? Alkaline phosphatase is dangerous or it is harmful for the liver if we can consume that unpasteurized milk. Alkaline phosphatase can damage our liver, which is unsafe for human to consume that type of milk. Here in the place of the substrate taken such as an para-nitrophenol phosphate, here we can another option we can here as a substrate we can take disodium phenyl phosphate which is used in place of an para-nitrophenyl phosphate. Here in place of para-nitrophenyl phosphate we can take disodium phenyl phosphate. Here the para-nitrophenol phenyl phosphate will give yellow color and this disodium phenyl phosphate will give the blue color. Here if the blue color is been present, it will indicate the presence of the alkaline phosphatase enzyme in the milk sample. Alkaline phosphatase, it is dangerous or harmful for human, which will damage the liver in the human body. What are the disadvantages of this reaction or this test? It will give the reverse reaction. Reverse reaction means the product will be converted into the substrate and substrate will be converted into the product. This is known as an reverse reaction. Here if in case the alkaline condition is not been maintained or any temperature such as an parameter such as an temperature, its molarity or its buffer solution, if this is not been properly maintained, the reverse reaction is been taking place. Here the chances of reverse reaction is been there. Here if the reverse reaction takes place, the product is again converted into the, or it will again form the substrate which is an para-nitrophenol or disodium phenyl phosphate, which will here reverse reaction means the product will be converted into substrate or substrate will be converted into the product. This is all for this alkaline phosphatase test.